Hey everybody, and welcome to the last lecture covering the urinary system. So this will be the last video lecture before exam 3. And I want to start off by talking in a little bit more detail about the processes going on within the nephron of the kidney, which is reabsorption of material and secretion. So if we take a look at the nephron, it's this series of tubules, starting with the proximal convoluted tubule, the loop of Henle, moving on to the distal tubule, and the collecting duct. So that is the pathway that filtrate is taking through the nephron. So you're going to have filtrate that's been removed from the blood through glomerular filtration. And so that's going to be passing through the lumen, or the hollow space in the nephron. And as that filtrate moves through the nephron, there's going to be material exchanged between the filtrate and the interstitial fluid. And material that's put into the interstitial fluid can then diffuse back into the peritubular capillaries that surround the nephron. So this is how anything that's been filtered out of the blood that needs to return to the blood is returned, is through this process of reabsorption. So you have, as filtrate is flowing through these tubules, there's going to be material that can pass through the epithelial cells that make up those tubules. And you've seen here this absorbing surface has those microvilli in the lumen of the proximal tubule primarily. So there's lots of reabsorption going on here in the proximal tubule, as we went over last time. But there's a couple of different ways that material can be reabsorbed. And that the differences between these two are how it gets out of the nephron, how these materials get out of the nephron. So one option is called the paracellular route. So any material that is in the filtrate can pass between spaces between the cells. So these cells are not continuously joined together. There are some spaces in between the cells that make up the nephron, the epithelial cells of the nephron. So this is the paracellular transport, where a material simply diffuses through these gaps between the cells. And paracellular transport is going to move substances along their concentration gradient. So if there's a lot of a substance here in the filtrate, it can diffuse passively. So that means that there's no use of ATP for the paracellular route. So this is a passive process that requires no energy. Now the other way that substances can move, and before we go on, these paracellularly transported substances are going to be like your small things that can diffuse readily. So your electrolytes can go by the paracellular transport pathway, as well as water can move by osmosis. And the other way that materials can be transported is through the transcellular pathway. And how this occurs is the same as transcytosis, that when we studied cellular transport, is you'll have a substance being taken into the cell across the plasma membrane, and that substance is basically spit out the other side through the basal side. So the apical side of the cell faces the lumen. The basal side is going to face the interstitial fluid. So in this transcellular pathway, the cell takes up, sometimes actively, sometimes this uses ATP, if a substance has to move against its concentration gradient. So this can also be a passive process, but in some of the specific examples that we'll talk about, transcellular transport does require ATP in a lot of cases. And this transcellular transport, using ATP in many cases, leads to the fact that the kidneys use up a lot of energy. About a fifth to a quarter of the ATP in the body is directly used in these transport pumps to pump things out of the filtrate and reabsorb certain substances using ATP. So transcellular transport is going to get some of your bigger things like glucose, amino acids, and also things that require pumping against their concentration gradient. So those are the two types of transport that can lead to reabsorption of a substance, meaning a substance made it into the filtrate, but it's going to go back to the blood. And your body makes about 180 liters of filtrate every single day. So obviously we don't lose 180 liters of water every day. So we lose about 1.2 liters of water a day. So that means that the vast majority of water that's being filtered out of the blood is getting returned to the blood. As well as about 99% of things like electrolytes, glucose, amino acids. Most of the solutes that are filtered out of the blood are going to get returned to the blood. And the reason that this filtration takes place at all is just so that the body has a way to fine-tune concentrations. But as I mentioned, as you can see from these numbers, there's lots of transport occurring to reabsorb substances from the filtrate. And of course, these transport mechanisms, depending on what you're talking about, can be modulated by things like hormones. So we'll talk more about hormone effects on the process of reabsorption, particularly with water. So the other type of transport that's going on within the tubules of the nephron is secretion. And secretion is the opposite of reabsorption, meaning instead of a substance going from the filtrate back to the blood, secretion involves a substance being transported from those peritubular capillaries back through these cells and into the filtrate. So the blood is essentially secreting these substances, and these cells are secreting those substances into the filtrate. So things that need to be further removed from the blood. And why is this necessary? It's because some wastes are not 100% filtered out in the glomerulus. Meaning after glomerular filtration, there can still be some waste left in the blood that needs to be removed, that didn't make it out into the filtrate. And how that waste is removed is these peritubular capillaries basically eject it, and it's able to be transported into the filtrate and out eventually into the urine. So these are the three basic transport processes that are going on. Two transcellular and paracellular transport in the case of reabsorption, and then tubular secretion is going to involve movement of substances out of the peritubular capillaries and back through these cells back into the filtrate. And I'd also like to mention that secretion is always going to be an active transport process. So when you're secreting waste, you're putting more solute into a place that already has a high concentration of solute, most likely. And so that's going to require ATP use. So secretion from the peritubular capillaries back into the filtrate is going to require ATP. I'd like to talk about specific ways mechanisms of transport for certain substances. And in the proximal tubule, this is incredibly important because the proximal convoluted tubule is where much of the reabsorption, particularly for things like nutrients and water, is occurring throughout the nephron. Some of the most important reabsorption processes are going on in the proximal tubule. So first I want to talk about how substances like glucose and amino acids are able to be transported from the lumen, where the filtrate is, of the proximal tubule, across these cells and back into the blood. How do you reabsorb glucose and amino acids? So we've got glucose here in the lumen, and this is a big molecule, and we're trying to move it. So it's going to require actually secondary active transport. So you might have to think way back to last semester when we learned about different transport mechanisms. And secondary active transport relies on building up a concentration gradient of one substance and using flow of that substance to transport a second substance indirectly. So that's secondary active transport. So let's take a look at how that works. In these proximal tubule epithelial cells, there are lots of sodium-potassium pumps. And what you may remember from looking at neurons is that these pumps are antiporters. They're going to split ATP, so this is an active process. And they're going to pump potassium into the cell, and at the same time pump sodium out of the cell. So you're going to be building up large concentration gradients of sodium outside the cell in the interstitial fluid, and also a concentration gradient of potassium inside the cell. But we'll disregard potassium for a second. So you've got this big concentration gradient of sodium, and there's actually going to be sodium in the filtrate as well. So many electrolytes make it into the filtrate, and these cells are pumping their sodium out. So on both sides here of the cell, you've got a concentration gradient. There's more sodium outside the cell than there is inside. So if we were to open up a channel, sodium would want to rush into the cell. So we've built up this concentration gradient using an active process, pumping. So what you have here is going to be a carrier protein that's going to allow sodium to diffuse into the cell. Now this particular carrier protein, once it's active, it's going to take sodium into the cell, and that rush of sodium is going to carry large molecules like glucose, amino acids, etc., into the cell. So that is how glucose, these large substances like glucose and amino acids, can make it into these cells. And once they've done that, they build up in greater numbers inside the cell, and then they can go through facilitated diffusion. So they build up in concentration, they travel with their concentration gradient into the interstitial fluid, and then back into the blood. So that would be how a substance like glucose and amino acids are transported during reabsorption in the proximal tubule. And almost 100%, the vast majority of any nutrients that get filtered out of the blood are going to be quickly reabsorbed in the proximal tubule. The next substance I want to talk about in terms of how it's reabsorbed is the bicarbonate ion. So what you may remember about bicarbonate is that it's an important buffering ion that's going to soak up hydrogen ions and potentially release them in order to buffer changes in pH. So pH is all, again, about the concentration of hydrogen ions in solution. And this bicarbonate ion can absorb hydrogen ions to fight decreases in pH or release hydrogen ions to fight increases in pH. So this is a great buffering system, and this ion is going to be critical for our body's ability to maintain acid-base homeostasis. And there's lots of bicarbonate in the blood, and it's a relatively small molecule, so lots of bicarbonate is going to make it into the filtrate of inside the proximal tubule. So we need to reabsorb that bicarbonate, and how that happens is kind of a roundabout, it's a mechanism that seems a little too complicated, but I'll try and break it down and help you understand something about the process. So you have this bicarbonate out here in the lumen, in the filtrate, and we need to get it back into the blood. So how that first occurs is that there is a sodium hydrogen ion symport pump, or excuse me, antiport pump. So sodium is being pumped into the cell, and hydrogen ions are being pumped out of the cell. And when a hydrogen ion meets bicarbonate, it's going to convert to carbonic acid, H2CO3. So there's an enzyme called carbonic anhydrase that can catalyze, it's an enzyme that catalyzes two reactions, one reversible reaction. So the substrate for this reaction is going to be H2CO3, that carbonic acid that we just formed out here. And carbonic anhydrase can convert carbonic acid to carbon dioxide and water. So that's what is going to happen here. Carbonic anhydrase makes CO2 and H2O out here in the filtrate from, the hydrogen and the HCO3. So the ultimate products out here are CO2 and H2O. Now once CO2 and H2O have been formed, those can diffuse into the cell. So now we've effectively taken these molecules and moved them into the cell, just in a different form. So now you have CO2 and H2O, and carbonic anhydrase is also going to be located inside these cells. So it's going to perform the reverse of that first reaction, and turn these two back into H2CO3. When this happens, H2CO3 is going to convert back into HCO3 minus, our original bicarbonate ion, and our hydrogen ion. So the fate of the hydrogen ion is to be pumped back out of the cell again, where it can pick up another molecule of HCO3 minus. So hydrogen is kind of going to go in this cycle where it's pumped out of the cell, picks up a bicarbonate, goes back into the cell, released again, back out of the cell. So hydrogen ion just kind of circulates like this. But each time it's going to effectively bring in a molecule of bicarbonate. So what happens to that bicarbonate eventually is facilitated diffusion back into the bloodstream. And so this will be the carrier protein facilitates the diffusion. This does not require ATP, whereas this hydrogen ion sodium antiport pump does require ATP. So somewhat complex, but requires carbonic anhydrase and playing a game of fetch with hydrogen, where the hydrogen is fetching the bicarbonate ion. And this is ultimately going to bring bicarbonate out of that filtrate back into the blood, where it can again serve as an essential buffering system to help us avoid changes in pH doing any damage. So the last type of reabsorption that I would like to discuss in terms of the proximal tubule, which is again doing a vast majority of the reabsorption in the nephron, is water reabsorption. And in the proximal tubule, we call this obligatory water reabsorption because there's no effect from hormones. So this type of water reabsorption is always occurring at the same rate and in the same way. So there's no fine-tuning of water levels going on in the proximal tubule. This is simply just going to be reabsorption of water under all circumstances. So what we've talked to up to this point is you have solutes out here in the filtrate, things like glucose, sodium being reabsorbed. We've shown sodium pumps bringing sodium across, amino acids, waste, other things. So you have movement of lots of solutes into the cell and then back out of the cell and into the blood. So as solutes move, what you'll remember from talking about osmosis is that water follows solutes. So wherever solutes have higher concentration, that's where water is going to go by osmosis. So as all of these different reabsorption processes, the ones that we've talked about previously, are occurring, movement of those solutes in this direction is also going to draw water out of the filtrate. So about 65% of the water that's reabsorbed in the entire nephron is going to happen through this obligatory water reabsorption. And there is some diffusion that occurs, and it's going to happen primarily, diffusion of water happens through channels called aquaporins. So we'll talk more about aquaporins in terms of hormone regulated or facultative water reabsorption later. But in either case, aquaporins are channels in the protein that allow water to diffuse. And it's, again, going to follow the solutes that are being transported. So moving on to the nephron loop and what's happening here. In the nephron loop is where you get about 20% of the water reabsorption that occurs in the nephron. Remember, 65% of that is happening here in the proximal tubule. And as you move into the nephron loop, you have these two ends of the loop, the descending limb and the ascending limb. And these two are very different from one another. You can see one difference is the thickness of the membrane. In the ascending loop, it gets a little bit thicker. It's got those cuboidal cells, as opposed to the squamous epithelium that makes up most of the descending loop. Now, what this leads to is going to be lots of water reabsorption happening here in the descending limb. So the descending limb is going to be permeable to H2O, meaning that water can leave the descending loop. And remember that the loop of Henle is going to be located in the renal medulla, which I mentioned is very salty. So there are lots of dissolved solutes down here in the renal medulla. And the further down you get, the closer to the end or the bottom that you get in the renal medulla, the further down you get, the saltier it is. And so the more water reabsorption can take place. Because the more solutes you have, the higher your osmotic pressure. So there is pressure pulling water in this direction. But that also requires that the descending loop is impermeable to salt, or impermeable to electrolytes. So in the descending loop, electrolytes cannot be reabsorbed. So as you go further down the loop, water is being removed, is being reabsorbed, and that leads to an increase in the concentration of solutes, like those electrolytes, the further down in this loop you get. So as you're traveling this direction, you're removing water, it's being reabsorbed, and the concentration, because you're removing water, the concentration of electrolytes in the loop are increasing. And remember, those electrolytes cannot leave this descending loop. Descending limb of the loop is impermeable to electrolytes but permeable to water. So now, on the flip side, on the other side of the loop, you have the ascending loop, which is impermeable to water now, but permeable to electrolytes. So in this ascending loop, you actually have symporter pumps that are pumping sodium, potassium, and chlorine ions out into the interstitial fluid. So there is going to be reabsorption of ions occurring here in the ascending loop. So what you have is water being reabsorbed here in the descending loop, sodium, potassium, chloride ions being pumped out into the interstitial fluid in the ascending loop. And what that is doing is, because the concentration of electrolytes is increasing the further down you get, means there's a high concentration of these electrolytes to be pumped out in the ascending loop. And as they're pumped out into the interstitial fluid, that's going to increase the osmotic pressure to draw water out from the descending loop. So this is called the countercurrent multiplier. It's countercurrent because the direction of the current in the descending versus the ascending is opposite. You have the current flowing down in the descending and up in the ascending loop. So there's a countercurrent here. And the multiplier comes from the fact that because these two are close together, as you travel down lower, the concentration of solutes is going to increase. These are going to multiply one another. The exit of electrolytes here is going to pull more water out here. And as you get lower and lower, you get higher concentration of electrolytes within the nephron. So what this countercurrent multiplier is helping to do is to establish this salty environment that's going to allow water reabsorption to occur in the medulla. Now, another thing to note is that it's also going to be salty near the collecting duct. The collecting duct is going to exist in the medulla as well. And as we'll see in the near future, there's going to be water reabsorption occurring here in the collecting duct and in the distal tubule as well. But this is not going to be your obligatory water reabsorption as we saw in the proximal tubule, but it's going to be facultative. Water reabsorption here in the distal tubule and the collecting duct is going to depend on the presence of hormones. So I'd like to also take a minute to talk about another mechanism that's used to preserve this osmotic gradient that we've set up. So again, the osmotic gradient being those electrolytes that are being pumped out here are accumulating, and the deeper you go in the renal medulla, the more salty the environment is, the more electrolytes are found in the interstitial fluid. So I first want to start by saying in the renal medulla contains tissues that still need a blood supply. And so the peritubular capillaries are going to provide that blood supply to keep the tissues out here healthy. And at the same time, you can probably imagine as a blood vessel is flowing through the renal medulla, it might tend to pick up some sodium ions So you would expect, based on your knowledge of diffusion, that sodium is going to go where from high concentration to low concentration. So you might think that sodium would diffuse into this vasa recta, and that would remove this osmotic gradient that we've established. But that's not actually the case. So what happens is, in the vasa recta, which is just another name for the peritubular capillaries that you see in juxtamedullary nephrons, we have a system called the countercurrent exchanger. And all this means is that as the vasa recta, which again is supplying blood to the tissue down here in the renal medulla, it needs nutrients and things found in blood to survive. So as you would expect, as the vasa recta goes down into the medulla, it is going to pick up sodium and chloride and potassium ions, and it's going to become more concentrated as it goes deeper into the medulla. And at the same time, it's salty out here, so you're actually going to lose water, and that's going to again contribute to an increased concentration here. So in this peritubular capillary, and blood is flowing this direction, filtrate is flowing this direction, the current of the nephron versus the vasa recta is counter to one another. Blood flow is going here, filtrate is going here, so there's counter current. And once you've got this concentration, this high concentration of electrolytes in the vasa recta, lower concentration higher up, and higher concentration the further down you go. Once you start to ascend in the vasa recta, now you have a high concentration of electrolytes, and that's actually going to favor diffusion of water in to the vasa recta, and those electrolytes that it picked up are going to be diffusing out. So essentially you're getting water for electrolyte exchange. Here you're picking up electrolytes and losing water, and it becomes more and more concentrated, and then you're exchanging again those electrolytes to pick up water here. So this vasa recta is basically going to maintain this osmotic gradient. On one side it picks up electrolytes and loses water, on the other side it gains water and loses electrolytes. So it balances itself out here, and is able to provide blood flow to the tissues that need it down here without disturbing this salty environment, this osmotic gradient that we've established here in the renal medulla. So the countercurrent exchanger is a mechanism to maintain that salty environment while still providing blood to the tissue in the medulla. So to wrap up looking at the structure and function of the nephron, I'd like to talk about the last part of the nephron, the distal convoluted tubule and the collecting duct. And these two are functionally similar, so they're often lumped together. They're involved with some electrolyte reabsorption, some secretion. So secretion of hydrogen ions in the collecting duct is one way that pH is controlled. But what we're going to focus mostly on is facultative water reabsorption. So as opposed to obligatory water reabsorption that we mentioned previously, facultative water reabsorption is going to occur as the body needs it. So in response to release of certain hormones. And this is going to be, the body has great control over facultative water reabsorption, and that's going to help us to be able to maintain fluid homeostasis. So up to this point in the nephron, in the proximal tubule and in the loop of Henle, you've absorbed, or reabsorbed I should say, between 85 and 90% of the water that's entered the filtrate. So what's left is, by the time you get to the distal tubule and the collecting duct, is about 10% of the water that you started off with. Now if you do the math, your body is filtering, the kidneys are filtering or producing about 180 liters of filtrate every day. So in a day, if we didn't reabsorb that last 10%, we'd be losing 20 or so liters of water. And of course we don't have enough water to go around. That would dry us up pretty quickly. So there is a mechanism for water reabsorption. But I will say that without hormonal influences, the cells that make up the walls of the distal tubule and collecting duct are impermeable to water. It's only when certain hormones adjust these cells that they're able to cause water reabsorption. So let's talk about first the cell type that makes up the walls here are going to be called principal cells. These are the primary cell type that you'll find making up the epithelia here. And what principal cells are remarkable in doing is responding to hormones. So they are responsive to hormones. And the first hormone that we'll talk about that they respond to is antidiuretic hormone. So ADH is going to be produced by the posterior pituitary gland. And it's going to, as the name implies, antidiuretic means that it's going to prevent water loss to the urine. It's going to make urine more concentrated, less water. So how it does that, how ADH exerts its effect, is by increasing the number of aquaporins in these principal cells. So when a principal cell is exposed to ADH, it's got receptors for ADH. It's going to manufacture and add more aquaporins into its plasma membrane so that it can transport water. So now, whereas before ADH was present, these cells were impermeable to water. So any water that comes this direction is just going to be lost to the urine. Now with ADH present, there are aquaporins that are going to allow water to be reabsorbed. So the second hormone that's going to have an effect on water reabsorption here is aldosterone. And what aldosterone does is it's going to increase the amount of reabsorption of sodium. So as you know, as we've kind of talked about ad nauseum in this lecture, water follows electrolytes. Water follows the solutes. So the more sodium that you're reabsorbing, the more water is going to follow it. So aldosterone, in increasing sodium reabsorption, is also going to increase water reabsorption. But this effect will only happen if ADH is present. Otherwise, there would be none of those aquaporins. So again, ADH has to be present for there to be more water reabsorbed here in the late nephron. So these two work together. ADH adds the aquaporins. Aldosterone is going to increase water reabsorption by increasing sodium reabsorption. ADH is going to be, again, produced in the posterior pituitary gland under the control of the hypothalamus. And aldosterone is produced by the adrenal glands that are situated right on top of the kidneys. Now, ANP is actually going to oppose the effects of ADH and aldosterone. So the way that ANP exerts its effect is that it's going to have an inhibitory effect on ADH and aldosterone. And so as it inhibits these two, it's going to increase the amount of water that's lost to the urine. So hormonal control is going to supply the ability to fine-tune the amount of water that's either lost to the urine or reabsorbed into the blood. And in doing this, we can control not just how much water there is, but by proxy, we can control blood pressure and the concentration of solutes. So that'll do it for our lesson on the kidneys. And good luck on your exam.